You know, thank you everyone for coming in person and online for the dissertation defense of Benjamin Gincley. A few words of background. Ben was an undergraduate student at Northeastern University when I was an assistant professor. Ben was enrolled in the bioengineering program with Zach. Zach, you're there as well? Online? Yes, you are. Okay, cool. So you can see those who are in person, you can see Zach over there. So we initially started working with Zach on what was a little tiny bead embedded inside a wooden box. You remember printing that, Zach? Yeah. And that was to develop a low-cost microscope of sorts. And so we worked for some period of time, and then I had the opportunity to pitch a design project as a capstone project for in bioengineering at Northeastern University. And even though I was in civil and environmental engineering, the department there was quite generous to allow me an opportunity to share some of the projects that we had to offer. And it was a very sort of I remember making that presentation. It was about 5 minutes, 5-minute long pitch. And I think that presentation turned out to be a really, really important one, right? It was a 5-minute pitch to a class full of undergrads. Because during the group that picked up that design project of building this low-cost thought process of building this low-cost imaging system, Ben, Zach, Hannah, Gabe, Shiraj, and Kyle, yes. So 6 of them got together, and over the course of the semester, they designed what would be the first prototype of the device that Ben is going to talk about today, and they ended up winning the capstone design competition. So which was fantastic. Somehow during that period, I managed to talk Ben into staying on to do graduate research with me. Again, you talk about getting lucky. I got lucky with a great capstone design team, and then I got lucky that Ben decided to stay and work with us. So Ben then worked on this project for a couple of years at Northeastern University, and then moved to Georgia Tech, where he started his PhD. And what you'll see today is a lot of work that has gone, and as you see the presentation today, and as Ben conveys it to you, it almost seems very simple, but it's simple because there is a lot of great engineering that has gone behind it, so that it is a product that is almost ready to go out into the world. It is already out in the world right now. So I'm very proud of what Ben has accomplished over the course of his PhD. He has not only put out high quality manuscripts out there, he's collaborated with a diverse group of people, is co-author on some very nice manuscripts out there as well. But more importantly, I think what Ben has done over the course of his PhD is build a pretty robust network across the algal industry. And this is because he took leadership, volunteering with different organizations. And I think, I think that's going to serve him really, really well in the long term. So we're going to sit here for about 40 minutes, listen to him share all the wonderful things that he has done. After that, there is going to be a period of 10 to 15 minutes where there is going to be an open Q&A for the public. And then we're going to have to ask everybody except the committee to step out of the room. So with that, I'm going to hand it to Ben.
All right, yeah. Thank you. All right, so I'm going to start sharing this presentation.
So if you're online, can you see the presentation? Yes,
yeah, cool. Okay, well, thank you, Amit, for such a great introduction. I don't think I could say anything else beyond that. So why don't we just jump straight into it and not waste any time. So today we'll be advancing the state-of-the-art for affordable flow imaging microscopy with ARTiMiS. All right, so we'll go through a little bit of background first. When people first hear the word algae, I think most people often think of this, pond scum, right? If you look at them under the microscope, microalgae actually look like this. They're tiny cells. Or specifically, this one type of algae called Nanochloropsis looks like these tiny circular cells. Microalgae is actually a very broad term, and it encompasses multiple kingdoms of life. There's some 50,000 known and documented species of different kinds of algae. And they range from being single-celled organisms to living in colonies or filaments. And they can be made of simple materials like just a regular cell membrane that you might have learned about in high school biology, all the way to really intricate silicate-based exoskeletons, which are some of those diatoms that you see there. So they're very cool to look at. So microalgae are interesting biologically, but the question is, why are they important for us people, especially in our modern day? So microalgae can be used for a lot of industrial products. They're used to produce biofuels, to produce pharmaceutical products, feeds for agriculture and aquaculture. They're also used in a lot of human health supplements. So if you've ever seen a vegan omega-3 or a DHA supplement, that's made with algae. And as an agricultural crop, algae are really important because they can be grown on non-arable land, so like in the middle of the desert. They can use non-potable water, in other words, salt water or brackish water. They're really efficient at capturing carbon. And when you grow them at industrial scale, they use a fraction of the aerial footprint of traditional ag. So that makes them very attractive for the 21st century of agriculture and bio-based products. And if you've seen anything like spirulina or chlorella or astaxanthin on store shelves, that's algae. So it's important to measure algae in order to understand how we can improve these processes. Engineers love control systems, and there's really two main types of control systems. There's open loop systems and closed loop systems, or as you could put another way, the just wing it, set it and forget it systems, and measure and adjust systems. And we, of course, prefer the measure and adjust systems. So it begs the question, how do people currently measure and adjust microalgae today? So in order to answer that question, I interviewed dozens of different practitioners across multiple fields. This includes surface water resource management, all the way to everyone working in industrial algal biotechnology. And there were a few key technologies that kept coming up as being some of the most common tools that people use. The first is using a manual microscope, and this is not too different from what you might have used in middle school or high school biology class. There are advanced automated microscopes that are very high throughput, very sophisticated. And then there's a number of molecular techniques like qPCR, if you've heard of that, to do deeper analysis of some of these algae. Focusing specifically on the imaging technologies, there's a range of different options currently available. And this is the current state of the art. So they range from everything from do-it-yourself projects that come out of academia that can be done for relatively inexpensively, but they require you to build the entire product yourself, all the way to really expensive, really high-tech devices. So seeing that these were the options and this was the spread, it was a little curious to me at the time when we first started working on this project, because recently in 2019, there had been a lot of advances in microfluidics, in low-cost credit card-sized computers, in new optics, courtesy of the cell phone industry and getting really good phones and cameras in your pocket, and data-driven automation. So machine learning had really just come onto the scene at that time. And so it was interesting that there wasn't any product that was incorporating all these and was also available on the market. So we're going to quickly walk through the different studies that we're going to talk about and how those different factors are going to be relevant in just a second. So first, we're going to go through the development of a novel flow imaging microscopy instrument, that's ARTiMiS, how we developed it and then evaluated it. Then we're going to talk about an extended sampling campaign that we used ARTiMiS to study a wastewater treatment system. And finally, we're going to look at a novel methodology using deep learning to get new insights through cell phenotyping. All right, chapter 3, introducing ARTiMiS. So before we talk about ARTiMiS, we need to understand why ARTiMiS even exists in the first place. So, like we talked about a few minutes ago, in 2019, people really had just two options if they wanted to look at algae under the microscope. You could use a benchtop microscope manually, or you could use a commercial instrument like the flow cam. The benchtop microscopes were relatively affordable as far as lab equipment expenses goes, but they were not affordable in terms of time. Running a sample manually took usually 45 minutes to an hour based on the people that I interviewed, of very mentally taxing and visually stressful work, meaning like it's tough to look at a bright light with your eye for an hour at a time 4 or 5 times a day. So the alternative to solve that problem is the instrument like FlowCam. However, most people characterizing algae or studying algae cannot afford FlowCam because they're so expensive. So really you had this choice. You could do capital efficient, money efficient, manual microscopes, or time efficient imaging flow cytometers or flow imaging microscopes. The question was, why can't we have both? Why isn't there an option that is both cost efficient and time efficient? So that's where ARTiMiS comes in. Not finding, excuse me, not finding a solution on the market. We did what any self-respecting engineering team would do, and we invented our own. So ARTiMiS is unique in that it offers really high quality images. It can do data processing on the instrument, which a regular microscope can't do. It's designed with user accessibility first and foremost in mind, and so it's meant to be really easy to use, both from a sample processing and a data analysis standpoint. And most importantly, it's available at an order of magnitude lower cost than something like a FlowCam that we just talked about. So in a nutshell, how does ARTiMiS work? Essentially, we take an aqueous sample, a water sample that has anything that we're interested in, could be algae, could be microplastics. We run it through the ARTiMIS instrument. Inside ARTiMIS, it will shine a light on the sample and it'll project that image onto a camera. We're going to take a lot of pictures of the sample as it's flowing through the instrument, and then ARTiMIS will convert that into some usable data that we can use, for any sort of analysis. So if you'll indulge me for a second, trip down memory lane, this is one of the very first images that we ever captured on an ARTiMIS prototype, going back to, like Amit had mentioned, those capstone days. So this is E. coli bacteria. They're about 2 microns. And while the quality of the image is okay, it's not great, especially by our modern standards for this product, it was really powerful at the time because, one, we could see something so small with an instrument that costs a couple $100. And 2, it was a microscope that was built by people who've never built a microscope before. So that was just really fun. So we can see small things on ARTiMIS, but the question is, how do we get more detail out of that? How do we turn that into useful information? So we're focused here on specific events. Events is anything that appears in our field of view. And we want to isolate them from the background in order to study them more closely. We ended up developing 2 different object detection algorithms to do that. One's called the fast detector, one's called the segmenter. They're essentially optimized for different types of samples. And so, when you're running the ARTiMIS, you have the choice to use either the fast detector, which is good for small single cells, and segmenter, which is better for more complex samples. So with object detection, we're able to take a very complex, very information-rich wide field frame like this, and we're able to extract meaningful information out of it in the form of these regions of interest that we can then study individually, and then eventually hopefully aggregate them and perform some sort of summary analysis. So we can see small things. How do we know if the measurements are accurate or not? In order to assess this question, I started testing with calibration targets of known sizes so that we can not only quantify how small we can see things, but also if there's any mistakes that the instrument is making when we just take the raw data essentially. And so this allowed us to determine the detection resolution, the empirical magnification of the optical system, which again was made out of lenses that cost like $10, and any measurement offsets that were needed. So for example, really small particles, like smaller than 2 microns, typically appeared larger than they physically were. And so there were offsets applied to those types of objects. So knowing how we can take the measurements and knowing how small we can see, in other words, about 2 microns, the next question was, how dilute can the sample be and us still get an accurate measurement? So here I'm benchmarking against a flow cytometer, which is a very sensitive instrument that can go across a whole range of concentrations to serve as our reference gold standard. And doing this type of test, we found that there is a specific limit of detection and limit quantitation. In other words, how small can we find things and how small can we repeatedly get a reasonable measurement from them. And so this allowed us to determine the effective range for ARTiMIS, which is about 10 to the 4 to 10 to the 7 cells per milliliter, which probably doesn't mean a lot to a lot of people. Essentially, this is ideal for industrial type samples, so cells that are growing in a big process. It's not ideal for surface water samples. And so at the end, I'll talk about Farhan. He's really optimized the system for that type of sample. Okay, so we can get total events, in other words, how many total cells there are in a sample, and that's useful. But it would be really nice if we could also tell them apart and say there's this much of this thing and this much of this other thing. So with the calibration samples, we know what size they are. So we're also able to develop a classification model using machine learning to identify and discriminate between the different particles. So what I found in the beginning is that particles that are very different in size, so let's say like a 6-micron particle to a 15-micron particle, 100% classification accuracy, which is what we'd expect. They're very different, and if we're making mistakes there, that's the problem. But what's more interesting is that there were errors with the small particles that were closer together in size. And so the question was, why are we having errors with these particles specifically? It turns out that the accuracy of these measurements is very dependent on how in focus a particle is. So on the bottom left here, you can see examples of images of the same object when it's in focus versus out of focus. And in the plot here, these long tails are demonstrating basically the noise in the measurements when objects are out of focus. So this is important to note moving forward. We need to have some way to filter out these out of focus events in order to keep our measurements as accurate as possible. And so just for reference, as we move forward, all the results that we'll look at later, they'll most of the time include this pre-processing step that will filter out of focus particles, basically have a classifier that says it's in focus or out of focus, and if it's in focus, it moves on for the next step. Okay, so if degree of focus is causing us this much trouble relatively, and the particles are of a specific size and they're very tight tolerance in the machining, biological variation could pose an even more significant problem. So potentially, surprisingly, to us at the time, with the right tuning of our classification models and the way that we were treating the data, pre-processing it, we were actually able to handle this biological variation reasonably well and get good accuracy when we're classifying the difference between two important microalgae species, chlorella and Cenodesmus. And so they're similar in size, they have a slightly different shape or morphology, and we're able to tell them apart pretty accurately. So this is a good sign for ARTiMiS. So just to kind of summarize everything that we talked to up to this point, we talked about the design of ARTiMiS and why it exists. We talked about some of the capabilities of ARTiMiS and how we validated its potential to take accurate measurements. And we demonstrated potential to discriminate objects, both synthetic samples and also real biological samples. All right, so up next, we're going to start looking at how we can use this in a wastewater treatment process. But first, this is the problem that we're going to be facing in this next chapter. How do we solve this more challenging classification task of it? Now it's not just a small circle and an oval, but now we have tons of variation. Even cells of the same genotype or the same type of taxonomic type have a lot of biological variation, different types. So this is going to present a lot of problems. And this is what we're going to work on in the next chapter. So chapter 4, we're going to be looking at using flow imaging microscopy to characterize microalgae and a nutrient recovery process. In other words, using algae to treat wastewater. So just a bit of background real quick. Microalgae are really good at treating wastewater. They have a natural affinity for some of the key nutrients that we try to remove in wastewater, specifically nitrogen species and phosphorus. Microalgae are also very efficient at low concentration ranges, which is really important for a lot of new state and local regulations that are tightening the limits of nutrient discharge. And so as a result, algae can be a more economical choice when it comes to designing new and improved wastewater treatment plants. So the site that we're going to focus on for the study is the Village of Roberts. It's in western Wisconsin. It's important because it's the first full-scale municipal wastewater treating algal process in the US, or one of the first. And so because of this, it's very important for us to study how the system works and understand what can we do better for future iterations of this technology. So in these systems, the key performance indicator that we're looking to focus on is how much phosphorus, or in this case we call it orthophosphate, is leaving the plant. we want to keep this basically at 0, and we want to know if it's above 0. So this is what we're focusing on. The objectives of this study were really twofold. First, we wanted to take an opportunity to compare ARTiMiS to the existing alternatives that were being used to characterize this plant already. And then what can we learn from a long-term study of this, and how can we apply that to the design of future plants? All right, so the existing ways to measure the total amount of biomass that includes microalgae and bacteria in these systems span, again, a range of different options. The most reliable and kind of easiest to get started method is manually measuring the TSS. It's relatively low cost, but you have to do it manually again. In order to automate that process and make it more convenient, TSS probes exist. So they're great because they can take measurements every 5 minutes and you don't have to touch it, but they also require a lot of calibration. And then finally, we're back to the FlowCam, which is a very high resolution instrument that can get really detailed data, but they're also very expensive. And so they increase the operation costs of these types of facilities. So we want to see, is it really good or is it not worth the price? All right, so comparing the results of measuring the total suspended solids, or how much total biomass is in these systems, we're comparing everything to the manual method as our benchmark. The TSS probe did a pretty good job, especially after we corrected for all the aberrations in the data. It's a good approximation. It's mostly linear. It's important to note there are some long tails, especially at the bottom of that plot. You'll notice that the probe was predicting values up to like 800 milligrams of TSS, whereas the actual reading was closer to 100. So when there are errors, they often are pretty severe. The FlowCam, which is very high resolution, precise instrument, surprisingly, while it's good at single cell stuff, it's not so great on a bulk scale. This had the worst error that we saw in the study, and it really just was not great for estimating how much total stuff was in the system. And maybe the last chapter was a bit of an indication or a prelude to this. We actually saw ARTiMiS having the lowest error of any of these measurements that we compared. And so it had the objective lowest error, the absolute lowest error, but also the error pattern was the most predictable of the three, meaning it's easiest for us to assign confidence intervals regardless of whether there's 100 milligrams or 800 milligrams. The confidence interval is pretty set, whereas for the other ones, like we saw with TSS probe, if it's giving a high number, it could actually be really low. All right, so fundamentally, FlowCam and ARTiMiS are both flow imaging microscopes. They take pictures. And so we can get a lot of really high resolution photos of the algae, and we can get a lot of them from both of these instruments. In order to compare the quality of the images they were collecting and the data that they were measuring, I took data from the FlowCam and data from the ARTiMiS and processed it in the same downstream pipeline for classification, which we'll talk about in a second. So it's the same classification structure. We're just comparing the two data types or data streams of the two instruments. Okay. The pipeline we're going to be using is we're going to take a collection of images of all different types, and we want to assign a class prediction to them. The way that we're going to go from images to predictions is we're going to use what's called a convolutional neural network. And essentially how these work is they will distill features from image data into a numerical representation. And the way they do this is actually very similar to how your own visual cortex encodes information from your eyes and turns it into what you see, hence the name neural networks. So these are convolutional neural networks. And anyways, when we get back to the comparison of the two instruments, we see that the classification accuracy between FlowCam and ARTiMiS is not that different. FlowCam does have higher accuracy, but ARTiMiS is not far off. And considering the price of the two devices, one being at least 10 times more expensive than the other, the value proposition of ARTiMiS here is pretty compelling. It's also important to note that while we have specific taxonomic groups, like we saw in the last slide, comparing like olive chlorella versus Monoraphidium versus Cynodesmus, within some of these key taxa, there are also different morphological types, or as we call them, morphotypes. And so they can range from being single celled all the way up to having 8 or more cells in the colony. And so the question is, like, why is this happening? And then also, does it matter? So in order to answer that question, we first need to be able to identify them. And the upshot of this is that the classification task of adding these additional classes does increase the challenge, but both instruments are still able to produce high enough quality data that we can classify them with high accuracy. So again, ARTiMIS is lagging behind the FlowCam data a little bit here, but still represents a respectable 92% accuracy. Okay, so with these classification data and with the high accuracy total biomass estimation from just a few minutes ago, we're able to construct these incredibly long, incredibly detailed time series. And by we, I really mean, like, I'm on the computer and Elaine, who's pictured here, is collecting all these samples every day for, it was literally like 3 years, over 1,000 samples. So this view that you're seeing now would not be possible without Elaine, who was on the call. So thank you, Elaine. But anyways, we get these really detailed pictures that have really never been seen before, because this type of high resolution has never been, performed quite to this extent and for such a long time. And so, what can we do with this besides making a really pretty visual? We can overlay some of the key performance indicators that we talked about before, how much orthophosphate is being removed from the system. And we can look for relationships between what the microalgae is doing and how the system is doing. We're specifically interested in these strings of black dots. That's when the system is performing really well and it's meeting its permit requirements, and the strings of red dots, where it's at risk of exceeding its permit and we have problems. So the summary of these results, a lot of data analysis went into this. When the system is performing very well, we see specific taxa and specific morphotypes of this taxa present. So I'm referring to small morphotypes of chlorella and Cenodesmus were most often associated with good performance, and the larger morphotypes of those species and a specific species of Monoraphidium were associated with poor performance. And so because of the nature of the analysis that we did, it allows us to look at how much large microalgae is present, for example, and predict in between 3 and 10 days that the system might perform poorly, so we can take corrective action ahead of time. So again, just to wrap up this chapter, we looked at the comparison of total biomass estimation between the instruments, we looked at the high quality images and how to classify them, and we looked at a really long time series and some of the insights we can get into optimizing these systems for better performance. All right, next up, we're going to talk about phenotyping.
You want to close that chat?
Oh, actually, yeah, I can do.
Okay.
Oh, it's
gone.
Okay, all right. Anyways, so label free cell phenotyping. So we talked previously about how different taxa, or in other words, different genotypes, can be typed into different, phenotype, morphotypes, and categories, right? We talked about 1 cell, 2 cell, 4 cells. But we also see intratype variation. So that, for example, this is a holotype, or in other words, a representative example of what a 1 cell chlorella looks like. But we also see a lot of different versions of this 1 cell chlorella. So again, the question is, does this variation mean anything? And can we determine some sort of structure from it? So that brings us to chapter 5, encoding the cell phenotype with some really cool deep learning methods. Okay, so just a bit of context. Again, we're looking at phenotyping as a technique. Previously, we had talked a lot about genotyping. That's determining what genus or family or species an organism is. Phenotyping is determining what it looks like, and the same species or the same strain of something can have vastly different phenotypes, meaning it can look vastly different and still be biologically the same thing. Just like people can look totally different. So that's phenotyping. The workflow is to do this quantitatively because it is kind of a qualitative subject. They're usually very time consuming. They require specialized expertise, and the instrumentation required for them is pretty capital intensive in order to get good results. So we had talked about flow cytometry previously a little bit. This is an example workflow of how you would do a phenotype assay with flow cytometry, and essentially takes a lot of work. There's a lot of design that goes into it. It's not easy to scale, and so it's challenging to adopt in the mainstream. So why does the phenotype even matter? In microalgae specifically, one of the key phenotypes for this class of organisms is lipid content. Lipid content and the macromolecule content in general is really important for essentially every product that algae biomass goes into. You want lipids high for biofuels, you want protein high for animal feeds, etc. And so knowing how much lipids are in the algae can tell us when it's important to harvest them and how to optimize process performance. Okay, so our question here, operative question, is it possible to use ARTiMiS to simplify this workflow and still get an acceptable result? Okay. The design for this experiment is very simple. We'll walk through it. We're taking one species of chlorella in a bottle, basically, and we're going to split it up into two different conditions. We're going to put it in the NR, or nitrogen replete condition. That's our control, not expecting anything to happen. On the other hand, we're going to put it into the nitrogen deplete, or ND condition. This is going to induce nitrogen starvation in the algae. This has been proven time and again to cause them to accumulate lipids very quickly. We have these two totally different phenotypes. We're going to use the flow cytometer to get a ground truth measurement of what this looks like. And then we're going to use ARTiMiS to see if we can approximate that measurement. So in the first batch, when we run this, we see a difference appear after a couple days, which is a good sign. We have the nitrogen deplete, that's the yellow and orange here, and the nitrogen replete is the cyan and blue, respectively. And after a couple days, we see a separation between the two, which is good. That means there's lipids in one and there's lipids in the other. Now, in the same study, we're going to repeat it. We're going to replicate it. Reasons for this are twofold. One, replicability of the results. And two, in production scenarios, it's important for your process to work batch to batch over time. And so we wanted to see if batch to batch over time, this method that we're developing is stable, or if it's going to have cause problems. So in the second batch, we see the same thing. We confirm this phenotype, that's the blue and the red line separating. And we actually see it separate a little bit earlier, and it stays separate the entire time. So it's important to keep in mind for a little bit. Now, while the means of these populations are far apart on the scales on the left, it's important to note that there is a lot of heterogeneity between these different conditions. There's cells that have low lipids that are in the high lipid condition, and vice versa. And so there's going to be some noise in these clouds that we're studying. Also important for what's coming next. Okay, so the question here is, how can we take images which have a lot of information in them, and how do we simplify them to basically a number or two numbers? The way that we're going to do that is with what's called a variational autoencoder. This is another deep learning model similar to the convolutional neural network that we looked at before. The VAE is comprised of two main components. You have an encoder and a decoder. The encoder you're actually already familiar with because we talked about convolutional neural networks. They're essentially the same thing. So we have a CNN that's going to take the image and it's going to crunch it down into a couple of numbers. The decoder part, that's new. But believe it or not, you might already be familiar with decoders. So if you've ever used some sort of generative AI model, you asked ChatGPT to draw you a picture, or you've used Stable Diffusion or anything, you've used a decoder. The part that you've seen is the prompt. So you write some words and you get the image, the output. So you're familiar with those parts. The part that is hidden to you when you let it spin its wheels is this conversion from words to numbers and then numbers to a picture. So this is fundamentally what the VAE is doing. We're just doing with pictures of algae. Okay, so in order to establish a good embedding, we do need pretty large training libraries to train these kinds of models. And conveniently, we already have an instrument that is capable of taking thousands of pictures and neatly organizing them. So we're in good shape. Okay, so batch one. Workflow is we take the images, we run them through the encoder, and this is that low dimensional representation, basically a coordinate, XY coordinate. After we train the model and we put batch one through this, we see a pretty diffuse cloud. The points are labeled by what sample they came from. And so immediately we can see there's a lot of noise, but there's a little bit of a structure to it. And so we see an increasing trajectory over time in the rightward direction. So in other words, there's something increasing with time that's good based on the design of the study. And we also see the conditions are pretty close in the first couple of days. And then we see them separate a little bit more in the later days. And that's really good because that's what we saw in the ground truth, the flow cytometry. Okay, batch two, same thing. Take all the pictures from batch two, put it through the encoder. This is the encoding, the 2D coordinates. Same thing, we see a rightward trajectory, rightward for everyone, trajectory. And also, more importantly, we see a more distinct separation between the two classes here, the two conditions, which is also what we saw in our ground truth flow cytometry. And remember, the flow cytometry data is measuring the actual lipid content, the literal lipid content. And so this is really good. So we have an orderly pattern to this coordinate space. It's a repeatable pattern, and it perfectly mirrors the ground truth. So these are all positive signs for using this method to do an approximation. Okay, so we looked at the encoder. Now what can the decoder do for us? So the decoder is able to generate images when you give it a coordinate. And so, for example, we can probe it for coordinate 1, 1, and it will show us what a picture of an algae cell will look like when it goes into spot 1, 1. And so this is useful because it can give us a visual map of what that cloud of points actually means, because just numbers is not very meaningful. But this gives us a collage or an overview of all the different phenotype variations seen in this experiment. And there wasn't any like annotation or labeling involved. This was a completely emergent property of the system, unsupervised learning. All right, so back to our original question for that fun detour. Can we use this VAE coordinate embedding to actually predict how much lipids there are quantitatively? All right, so I'll cut to the chase here. The VAE, while it was not the outright best method to predict the amount of lipid content, it was the next best method in rank of 2, 3, and 4 behind label-free flow cytometry. So this is using other measurements that the flow cytometer is able to record that don't require dyes and are not measuring the lipid content directly. There is one combination of pre-treating the data and using a specific type of fitting model where the flow cytometry comes out on top for the first one. But the VAE is the most stable and most consistent in that it's not really dependent on how you treat your data, what type of fitting you're using. It will give a good fit regardless. All right, so the wrap up of this chapter, we found that the VAE was very robust to characterize the phenotype, and it had a very good ranking of its different encodings and its fittings. We also saw that we could encode phenotype dynamics without knowing beforehand what we're looking for, because it's an unsupervised learning method. And then we also saw that they were stable across batches. And this is really important because that means you can train the model once on a calibration set of data and then use it in production for weeks or months in the future. All right, so let's wrap it up here. I just want to show you real quick, because Amit had mentioned in the beginning, this is a view of what ARTiMiS had looked like over the years. So all the way on the left there in 2018, that's the prototype of ARTiMiS when it was in Capstone. And over the years, there's been a lot of design iterations and improvements. Most of the pictures that I've shown in this presentation have been that hexagon version, so it's the 2022 variant. The current one looks a little bit different. That's the one in the top right, and that one is pretty much ready to go as a product to ship. So that's really exciting. All right, so just a visual overview of some of the chapter highlights that we covered. We talked about algae being a really important crop and some of the ways that you can measure it. We talked about why ARTiMiS was developed and how we validated it. We talked about a really interesting demonstration of ARTiMiS over a long time series. And we talked about some novel ways that we can try to determine phenotypes on a very affordable scale. So without dyes, without complicated workflows. And when you do a lot of work in a PhD, there's always more to do. I think they say that for every PhD, you spawn like 2 or 3 new PhDs worth of projects to do. So the work does not end today or in a month. There's a lot of improvements to user experience and accessibility that we're focusing on now that we've ironed out all of the technical aspects of how ARTiMiS works. We want to make it as usable as possible and as fun to use as possible. Just talking about some of the future work for some of the studies we talked about, we're going back to look at all that wastewater data that we examined and we studied to look for the impact of grazers or organisms that eat the algae, to see how that impacts the morphotypes, and then by extension, how that impacts process movements. And then I wish there was more time to explore other variants of the variational autoencoder family of models to characterize some of these more visually intricate taxa. So Senidosmas, for example, like we talked about before, can exist as 1 cell, 2 cell, 4 cells, 8 cells. And so what's shown on the plot here above is on the left is the chlorella. So that's all the data that we talked about. On the right is what the flow cytometry lipid measurement of semidesmus does. It's much more complicated. It's much more difficult to fit. Both the VAE and regression models do it. So that is something that if I can't do it, hopefully someone else can. So just to take it all home here, if you don't remember anything else from today, these are some of the key takeaways to remember. Algae are really important and useful crop, especially for agriculture in the 21st century. But in order to get to economic viability, we need new technologies to improve them. And hopefully ARTiMIS is one of those technologies. We developed ARTiMIS to bridge the gap between do-it-yourself projects that are really cheap and instruments that cost as much as a house in some cases. We demonstrated how ARTiMIS is as good as those really expensive instruments while still being a tenth of the cost. And we talked about some really interesting new ways that we can use modern advances in computer science to dramatically improve how affordable and accessible doing some of these complicated workflows can be. All right, so here's a list of some of the works that I've had the privilege of either leading or being a part of, mostly here because the committee cares about academic productivity. The work here wouldn't be possible without the funding support of these institutions, these agencies, and some of these competition programs that we've been able to take part in, as well as some of our key collaborators, most of whom are represented either in person or online today. So thank you all for supporting the work. And this is both my favorite slide and also the hardest slide for me. I write out what I'm going to say for every other slide, and like I don't read a script when I present, but I write it out as if I'm reading a script. For this slide, I can never write it. So in the top left, this is that capstone design team that won that capstone challenge in 2018. So they were everybody who started ARTiMIS as the version of the project that it is today. We wouldn't be here without everybody in Dr. Pinto's research group over the years. There's been a lot of undergraduate researchers who've given a lot of their time and attention and dedication and care to this project. So it also wouldn't be here without them. And you know, for people who are in a PhD or have done a PhD, it can get pretty tough sometimes, and having undergrads around really reminds you why you're doing what you're doing. So I'm forever grateful for them. Elaine, like I mentioned before, has been pivotal to collecting some of these more extensive data sets. Farhan is really the person who gives this project a buzz factor greater than 1. If you don't know what that is, you can save it for questions. But Farhan is really the technical lead, the lead designer of innovation for ARTiMIS in 2025. And so he deserves more credit than I can give. And of course, Dr. Pinto, I couldn't come up with anything to say, but like Amit had said, I wouldn't be in a PhD program at all if they hadn't invited me to be in a PhD. And I wouldn't have been able to go from Northeastern to Georgia Tech without Amit's support and patience. And so thank you, Amit. And that's it.
Awesome. So what we're going to do now is we're going to open this up for audience questions. Ben, can you close this slide and bring the stop sharing? And for the audience, please, if you're online, you can unmute and ask your question. If you're in person over here, please raise your hand and ask your question. So I'm looking online, I'm looking in the room. Ashwin had a question. So we'll take one from inside the room now. Ashwin, please.
Hi, thank you for the talk. And
Can you repeat the question for the online?
Yeah. So when you move from phenotype to genotype to phenotype, what were the challenges you faced? Because I'm assuming like it's one thing to discriminate different shapes, but then in the case of your phenotype, you're going to be like looking at different variations of the same shape, right? Were you able to still use the same workflow, or did you have like modifications?
Good question. So you're talking about going from like a species to the phenotypes we talked about in the last chapter, or the morphotypes?
The phenotypes of the last chapter.
Okay, so the question was, what is the challenge that you face when you go from doing genotyping or taxonomic analysis and doing discrimination between classes that way versus going to the type of cell measurement or determination that you can't use discrete classes for, the phenotypes of the last chapter that we talked about? Yeah, the CNN, like we talked about, so the CNN method of encoding visual information into numbers basically is effective, but the method of training the model, in other words, supervised classification, which we had done up to that point previously, it just wasn't possible. Is by design, it is a like a discrete quantized, list of classes, and it's either this thing or this thing or this thing, and there's no way it can be continuous. And so it's just discrete. And so we had to move to an unsupervised classification or measurement method with the variational autoencoder. And really the way that it is able to be unsupervised is that decoder part. That part is so key because it allows us to organically draw a map based on the data. And I don't have to go in and say, I want this coordinate region to be big cells, and I want this coordinate to be small cells, and these ones are fuzzy. So I didn't do any of that. The model training process itself does that. And so, yeah, we had to go from supervised to unsupervised methods to do that.
Awesome. Questions in person or online?
Oh,
I have a question.
Okay, Tim, yes.
So Ben, great talk and great work, by the way. Elaborating a little bit on that previous question that was asked, is there any sort of physical interpretability that you had to the two like parameters from your, I guess the latent dimensions in your variational autoencoder? Like one looked to maybe be related to size and one looked to be maybe related to fuzziness or something like that. But is there some way where this could be tied back to something with physical interpretability?
Yes, great question. I'm gonna share my slides again. Sorry about this. Okay, yeah, can everyone, you see the full screen version, right? Yeah, okay. Yeah, I guess it I'm showing now. Okay, yeah, so great question. I think everybody heard it. So we actually can do a more quantitative scoring of how the different latent dimensions correlate to physical parameters that the ARTiMiS measures. This slide wasn't included in the main part mostly because it was rendered for print, and so the text is kind of small. But if you squint at it, you can see that there are different specific measurements that have, like semantic meaning. So circularity is how circular it is, and there's a mathematical formula for that, et cetera. And so this is a Spearman rank order correlation comparing each of the two latent dimensions, that's the cyan and the blue respectively, against some of the features that ARTiMiS measures. And like you said, subjectively, some of them are like bigger and some of them are fuzzier, et cetera. So this lets us see, both latent dimensions are highly correlating just in the negative direction with things like edge difference. So that's how fuzzy the edges are. And then on the other side, they're positively correlating both in the same direction for things like area. So that's how big the cells are. So yeah, we are able to compare the latent dimensions to real measurements, quote unquote.
That's very exciting. Cool. Thank you.
Thank you, Tim. Awesome. All right, so questions? Over here, online. If you're online and you have a question, please, yeah, so Zach has a question. Zach just unmuted. Yes, please go ahead, Zach.
Thanks for such a stunning talk, Ben. It's really amazing to see the stuff. I was wondering if you saw anything interesting with the bacterial flocks in that time series. I know they were kind of like lumped into one category, but like, were there any kind of like differences in flocks over time that you saw?
Yeah, that's a great question. So that is part of the future work that unfortunately, with the scope of the projects, we weren't able to get into. I'd be really interested to study the shape and the size of those flocks and see if that had any implications for how things went. I think like you're alluding to, we ended up discretizing the different types of algae into different types, but we did not do that with the flocks. And the flocks range from really small to really big. And so it'd be good to do a more correlation type analysis between the size and process performance. When they're just bins as a whole of the total biomass, or like a fraction of the total biomass, it was a little bit surprising to see that they actually didn't have a huge impact one way or another. One of the limitations of this study is, although it was 2 years, there's a lot of stuff happening over those 2 years. And so any individual span of time was kind of its own unique thing. And so it was hard to translate results, but effectively the correlations came out to be basically negligible one way or another for everything to do with how many flocks or how much flock there was, which was surprising because I think before we did that analysis, part of the design of operating the plant was get the flocks down as low as possible and get rid of them. And they tried a bunch of things to get rid of them. And so that also, I guess, makes the interpretation of that result challenging because it wasn't an environmental observation study of like, let's see ecologically what happens organically, but this was an engineer manipulated system, or if the flocks were too high, like they literally got rid of them. And so that also messes with the results. But yeah, it's a very good question. I don't know.
It's totally okay to have that.
Yeah.
I don't know answer. All right. Questions from the room? Okay. Yes,
I've got
Georgia.
a silly question. I'm so sorry. So I'm interested in like the CAD process itself. And so when you started designing these prototypes, what kinds of materials did you use and how did you like specify what you wanted printed on what kind of material? So I know it's not very like technical, but I was just curious.
Yeah, that's a great question. Just because I don't know how sensitive the mics are. The question was, how did you design, or how did you choose materials, and how did you choose what designs to incorporate with what materials when you were prototyping? Because when you are prototyping, for people who haven't done this, you have a choice of like a plethora of different types of plastic specifically, in addition to non-plastic materials. To be honest, going back to like the core thesis of the why we did this in the first place, cost was usually the number one concern. And so we went with usually the most inexpensive, yet still mechanically responsive materials. So for people who do 3D printing, we use a lot of PLA, which is like basically the most basic material. We did experiment with other more flexible materials for specific parts. So there were individual components that being brittle and being like very hard was not ideal. And so we used other materials like PETG, which are more flexible in order to handle shocks. But there were other components that we needed to be rigid because at the end of the day, it is a microscope, and the optical components, if they're not exactly where they need to be, you were going to have problems. And so there are other parts where like we need this to be a rigid material. If it falls and like on the wrong way, it's going to break, but it shouldn't fall. So let's pick the hard material.
Thank you.
Awesome. Anyone online with questions? Yes, Jiju, yes.
I just have a question related to the full-scale waste, like algal wastewater. Is there any like correlation between the phosphorus removal with the different phenotype of algal algae that you are focused on during the research period? Like, is there any?
Yeah, so I think if I heard you right, is there a correlation between how much phosphorus is being removed and the types of algae that we see and the phenotypes of the algae? Yeah, so that's what that, like looking at, there's single cell chlorella, 2-cell chlorella, single cell cyanidesmus. So those ones were most associated with conditions where most of the phosphorus was being removed. In this type of system, we can't technically dictate causality because there's like literally hundreds of different parameters involved, and we don't have an experiment to prove one way or another. But the hypothesis there is that if the system is performing well and the algae are happy, they are dividing and they're growing really quickly. And it's been shown in literature that the larger phenotypes where you have 8 cells together, that's usually a stress response. And so the way we interpret it is if the algae are stressed, they'll form those clusters, those colonies. And then because they're stressed, they're not taking up as much phosphorus. And so in the preceding or the following week or so, the system won't do as well because the algae are stressed, they're not taking up phosphorus. Whereas on the other side, if they're all single cell and they're all basically happy, then they will continue to grow, the biomass will grow. And because the way they work is they take phosphorus into their biomass, they incorporate it into their biomass as they're growing. And then the way you remove phosphorus is you physically remove the algae from the system. So the algae are growing well, they're taking up the phosphorus. And that's how we interpret that result.
Thank you.
Yeah.
Great. Anyone online? Okay. If not, and in the interest of time, we're going to close the public section of the defense now. So thank you to everyone online and in person who joined for Ben's presentation. I'm going to ask those who are online to please log off. And for those who are in the room, please take a cookie before you leave.